Okay, let's go ahead and start. Anybody have any questions on anything up to this point? Okay, that's a good sign. We're gonna wrap up chapter 2 on chemistry and probably go ahead and start on chapter 3 on the cells this morning. So let me go ahead and open this up and get into nucleic acids, which is where we stopped the last time. And we can start now. Okay. We've gone through the organic molecules from carbohydrates, the sugars, the carbon, hydrogen, oxygen compounds. We've gone from there, we went into the lipids, which are also carbon, hydrogen, and oxygen based. The oils, the solid fats, the liquid fats, and steroids and we moved into the proteins. Excuse me, I have got this tickle that's been going on all morning. So anyway, we end up now with the nucleic acids. Nucleic acids are the DNAs, the RNAs, and certain molecules that we use to transport energy around our bodies, the ATPs. So the nucleic acids all use carbon and hydrogen and oxygen, but they're also going to use, there we go. Okay, now they're also going to use nitrogen for the amino acids and phosphorus for some of the chemical bonding. Nucleic acids are made up of monomers called nucleotides, and all the nucleotides have the same basic structure. The nucleotide will consist of a sugar, a ribose, or some sort of 5-carbon sugar that looks like this. So there's some sort of 5-carbon sugar, and on one side of the sugar, you're going to have a on one side of the sugar, you're going to have a phosphate group, and the other side of the sugar, you're going to have a base, and the base is a nitrogenous base. So all DNA or RNA molecules are assembled essentially the same way. This is a nucleotide, a phosphate group, phosphate, and a sugar, and a nitrogenous base over here. And so the 3 components then make up the nucleotide. And there are 2 types of nucleotides, one with the 5-carbon sugar with oxygen, and one with an oxygen atom missing. And so we say the one with oxygen is the ribose sugar. The one without the oxygen is the deoxyribose sugar. And so with these nucleotides, then we can create deoxyribonucleic acid, or DNA, or we can create ribonucleic acid, which is the RNA. And of course, we know that DNA is the basis for all of our genetic information. Okay, so here's a nucleotide, and it's a nicer drawing, but I think it's a little more confusing than what mine was. I tried not to make mine too confusing, but you can see here in the structure, you've got the pentose sugar, the 5. Stop that. Let me. Here we go. Now I'll try this again. Okay, here is the pentose sugar, the 5-carbon sugar right here. there's the phosphate group right there with one phosphate attached. And here is a nitrogenous base. And so here's a nucleotide, and here's a nucleotide, and here's a nucleotide, and here's a nucleotide. And our DNA molecules and our RNA use the sugar and phosphate as the backbone of the molecule. So you would have a sugar and the phosphate attached to another sugar and another phosphate, and so on. And this becomes the backbone of the DNA molecule. DNA molecule is a double strand that looks a lot like a ladder. And the DNA molecule, each DNA molecule is about 6 feet in length, and it's, you know, the thickness of a molecule. So it's very, very fragile and very, very long. And it has a unique shape. We don't see this double helix structure in many molecules at all, particularly ones that are that large. But every DNA is going to be assembled using nucleotides. So is RNA. The only difference between the nucleotide in the DNA is that it's missing one oxygen, and the nucleotide in the RNA has that oxygen. That's why we call it ribose or ribonucleic acid versus deoxyribonucleic acid. They're both 5-carbon or pentose sugars. Now, let's take a closer look here. Nucleic acids hold all of our genetic information. The DNA, in particular, is the blueprint. It's the cookbook for us. All of our genetic information is contained within our DNA. Every cell in our body has a nucleus. Every cell in our body that has a nucleus has DNA within that nucleus, and that is a complete copy of the entire amount of DNA for every or any cell. So a liver cell, a skin cell, a brain cell, a muscle cell, their DNA is exactly the same as all the other cells' DNA. It just, the segments that make them a muscle cell are turned on, and everything else is turned off. A liver cell has all the instructions on how to be a liver cell turned on, and everything else turned off, but the information is all there. Now, the DNA molecule itself is a double helix. It looks like a ladder, if you will. The shape of the DNA molecule is like this. It's shaped like a ladder, but a ladder that you could twist into a circle. So it's like a spiral staircase. The actual DNA strand is shaped something like this, with the bonding in it occurring like that. And that tends to give the DNA strand a little more strength and flexibility than just a straight chain would give it, you know, by being twisted like that. And so you've got these 6-foot-long DNA molecules made up of all these billions of nucleotides fitting together to contain our genetic information. Now, the key to the DNA molecule is the presence of the nitrogenous base that we find here. The nitrogenous bases, there are 4 different bases that are created from nitrogen in DNA. We have 2 called adenine and guanine. They're what are known as the purines. And then we have the pyrimidines. The pyrimidines are cytosine and thymine. So these 4 bases are what are the nitrogenous bases in the nucleotides for the DNA. And they will bind to each other. You know, you have that ladder-shaped structure here. You'll have a base here and a base here, and they will be held together with hydrogen bonds. And we find that there is a certain rule here for pairing these bases, and they pair what we call complementary. In other words, adenine will always pair to thymine. So here's your strand, there's your strand. And guanine will always pair to cytosine. You know, there's cytosine here, guanine there. You know, you could have, doesn't matter which side they're on. Guanine always pairs to cytosine and adenine always pairs to thymine because that's their complement, and they form hydrogen bonds between them to hold them together. And it's those hydrogen bonds that actually cause the molecule to twist into its double helix shape. Now, this is what DNA looks like right here. This is a DNA molecule, a segment of it anyway. This is the double helix. You can see how it has the spiral staircase effect. It has the complementary pairing between the adenines and the thymines and the guanines and the cytosines, for example. Here in green is cytosine. And I'm sorry, it got ahead of me. There we go. Let's try it now. Here in green is cytosine, and it's paired to guanine over here. You know, here is thymine, and it's paired to adenine. Always. You know, you'll notice that greens paired to yellows, they're not really green and red, they're not really red and yellow or green and blue, but you know, adenine always pairs to thymine, guanine always pairs to cytosine. Doesn't matter which position they're in. You know, if you have a cytosine on one side of the helix, you're going to be paired to guanine on the other side. That's just the way they assemble. And if a mistake does occur, for example, where somebody pairs where adenine paired to guanine, our DNA has enzymes that travel up and down the strands and look for mistakes like that and slice them out and replace them with the proper base. So when we say that it always pairs like this, it pretty much always does. Now, mistakes can happen, and mistakes will happen. But the error rate for a mistake here is something like 1 in a billion. So that's a lot of flexibility there for not making a mistake. So this is what the DNA molecule looks like. We have these bases paired up with each other. And let me say a few more things about this. A gene, bring my little marker back down. A gene is only the physical location on DNA for a genetic trait. The information on hair color or eye color or skin color or silly things like being able to roll your tongue or whether you have attached earlobes or dangling earlobes, or a little more serious, your blood type, for example, or your eye color or your hair color, your body structure. All of that information is contained in genes on our DNA. Now, the genes, again, a gene is nothing more than the location of that information on the DNA. So this could be a gene from here to here. That could be a gene, because the order of these bases dictates the what the gene will do. The order of the bases, you know, the adenines and guanines and cytosines and thymines lined up here. You know, here is starting here at guanine. There's cytosine, there's thymine, there's another guanine tucked in there. There's a thymine. You know, this sequence of bases determines what protein that that gene is going to make. And that protein determines what the gene's going to do. It could be the protein for hair color or eye color or blood type, or it can be a protein that's going to drive growth or reproduction or anything else. Our genes are nothing more than the physical location on our DNA for a particular trait. That's all it is. That's all what a gene is. The DNA is a complete list of all of our genetic traits. Everything about us, everything that makes us is contained, at least physically, is contained in the DNA. So let's move on now and see where this takes us. Okay, so what is RNA then? RNA is a single chain nucleic acid. RNA carries information from the DNA to the cell to direct the formation of a protein. The RNA is a single strand. It's a straight chain. Instead of being in a double helix, it's a single chain. It is made with a ribose sugar, meaning that it does have that extra oxygen. The only other difference is that it doesn't have thymine. It has a base called uracil. Other than that, other than the fact it's a single strand and it uses, it's a single strand chain, it uses ribose sugar, and it doesn't use thymine, it uses uracil. It's essentially the same as DNA with those exceptions. Now, we make 3 different types of RNA. We make what's known as messenger RNA. Messenger RNA carries the instructions from the DNA to the rest of the cell. The DNA never leaves the nucleus. The DNA uses RNA to do its work. The DNA says we need more hemoglobin. And so the messenger RNA will make a copy of the gene to make hemoglobin, and it will take it out into the cell from the nucleus, and it will use transfer RNA to assemble the amino acids in the order of the specified protein, because all proteins are made of amino acids in a proper order. Hemoglobin has 360 amino acids lined up one after another in a very specific order. The order of the bases in the DNA dictates the order of the amino acids. And so when the messenger RNA goes out to the cytoplasm, it has its instructions spelled out in what order the amino acids are going to be put together or linked together to make a protein. And the transfer RNA will start bringing those amino acids to be assembled. And the assembly takes place in the 3rd category of RNA, and that is the ribosomal RNA, because the structure where we make our proteins is called the ribosome. Let's see how this plays out for us today. Make sure we have our sound up. See if this works for us.
Units called nucleotides. One difference between DNA and RNA is that DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has 1 more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The 2 polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
I think we could say that the bases are the order is a little more than just significant. The order of the bases dictates the order that the amino acids will be assembled. So the gene is where we have all the instructions to make every protein in our bodies and to make us, essentially. So this is how the DNA genetic code works. It has the order of every base specified so that order pairs up, the order of the bases pairs up to a unique amino acid. And it's the hydrogen bonding here between the bases that cause this twisting of the DNA molecule into its characteristic double helix. Now, okay, third category here in our nucleic acids. And we'll talk more about DNA and RNA when we get into chapter 3 anyway. Now, third category, ATP, adenosine triphosphate. It is a nucleotide somewhat similar to RNA. It has a 5-carbon sugar. It has a ribose sugar here, but its difference is that instead of having 1 phosphate, ATP contains 3 phosphates. You know, here's your 5-carbon sugar with in ATP. You have your base over here, and over here you've got 1 phosphate, 2 phosphates, and 3 phosphates. The most important of the phosphates here is this 3rd phosphate. You know, this is a high energy bond between phosphate number 2 and phosphate number 3. This is why we call it adenosine triphosphate, 3 phosphates. This high energy bond here can be broken and restored. When we take ATP out to a site in our bodies where we need energy, we break that bond at the 3rd phosphate, we release the energy, and then we end up with what's known as ADP, adenosine diphosphate, and phosphate. And if we have energy available to us from eating something, we will reattach that bond and park stored chemical energy in there that we can use. Now, a couple of things about that. Let's go here to the next slide. And this is what it looks like. The 3rd phosphate, and you see the molecule there, there's the adenine, and there's the ribose sugar. When we have energy available from something we've eaten, you know, a donut or a piece of bread or an apple or whatever, we can take the calories in that food that we've consumed, and we can reattach the 3rd phosphate, and we can park energy in that bond. It takes energy to force that phosphate onto in the number 3 spot. How much energy do we have? About 7 kilocalories, actually about 7.2 kilocalories of energy is available. So it takes 7.2 kilocalories to force that bond back together, and we can park it there. The problem with that is that that ATP molecule will only last between 5 and 6 seconds, and then it breaks apart. So we make it and we have to use it right away. We can't store energy in ATP. We use it to transfer it from one, like to a muscle cell so the muscle can contract, or a nerve cell so the nerve cell can generate a nerve impulse, or, we use it to pump things across our cell membranes in active transport. We use energy all the time, obviously, but we have to keep making ATPs over and over and over again because they're only going to last between 5 and 6 seconds. Each one now has 7.2 kilocalories of energy. That's a fair amount of energy that we can do stuff with, but we can't store it. So, and what will happen though is when we break, as soon as we break that bond, as soon as we break that high energy bond, we release that 7.2 kilocalories of energy, and then we end up with adenosine diphosphate and what we call inorganic phosphate. If we have more energy available from our food, we can reestablish that. We can put that third phosphate back on, shoot it out to a muscle cell. It'll do something and it'll release its energy and goes back and gets recharged and does this over and over and over and over again. As long as we have energy available from food or stored energy from fat cells or from glycogen, we can keep reestablishing the energy parked in the ATP. We don't take energy directly from glucose to do things. We take the energy from glucose and we park it in ATP as a manageable unit, because there's a lot of energy in glucose. There's an awful lot of energy in a molecule of glucose, more than our cells can handle. So what we deliver the energy to our cells in is ATP. Glucose goes into the cells and we oxidize, we burn glucose and we make ATP. Our cells are busy making the ATP so we can use it that way. We can't handle the large volume of energy parked in a molecule of glucose at one time. We got to break it apart. And that's what we do with ATP. ATP is the currency of our bodies, how we power up all of our actions that require energy. So, okay, that is the end of chapter 2. And your lecture test on chapters 1 and 2 will probably become available next week, probably towards the end of the week. I'd like to go ahead. I want to get started on chapter 3 now. Chapter 3 will not be on the lecture test on test 1. Chapter 3 and chapter 24 are going to be on test number 2. Cells and metabolism, that's a whole separate test. Right now, we finished with unit 1, and I want everybody to get through their lab test first before I even think about opening up the lecture test. So probably won't open it till next week. And there is a study guide for this test, and I know that before next week, probably on next, let's see, what's today's Wednesday. Probably on next Wednesday, I'll do a brief review for you all. Just remind me, I'll do a brief review for the lecture test because I'll open the lecture test on Thursday. So anyway, any questions before I open up cells, chapter 3? Okay, then let me go open up that and go. Zoom. Let's see if I did this right. Okay, so everybody should see the slide that says the cellular level of organization, chapter 3. At least I hope so. Okay. So cells. Every living thing is made of cells. Doesn't matter if you are a one-celled bacteria or you're a human, you're made of cells. Every living thing is made up of living cells. A living cell is the smallest unit of life. There's nothing smaller than a cell that can be alive. Bacteria are cells, so they're alive. Viruses are not cells. Viruses are not alive. And despite the fact we're talking about killing viruses, a virus is nothing more than a strand of DNA in a code of protein. A virus looks something like this. Here's your DNA strand inside the, that's a highly detailed drawing of DNA, by the way, inside a protein coat, and it has structures on it to allow it to stick on a surface. It's not alive, that's a virus. Whereas a bacteria, whether it can be like a streptococcus with round bacteria or a rod-shaped bacteria like anthrax, for example, or a spiral bacteria like syphilis, those are bacteria. Those are all made up of cells, and they are alive. So we know that the cell is the smallest unit of life. And so, when someone says we're going to kill viruses, just nod your head and say yes, even though they're not alive to begin with. And the third thing is that living cells only come from other living cells. We don't create life with, in lab. There's no way to take chemicals and do things to them, like using large charges of electricity to mimic lightning strikes. There's no way to create life artificially like this. Living cells only come from other living cells. And this is really the whole basis of what we call the cell theory. Living cells come from other living cells. Cells are the basic unit of life, and every living thing is made up of cells, whether you're a plant or an animal or a fungus or a bacteria. We are all made of living cells, and we reproduce using cells. Now, in our bodies, we have over 200 different types of cells. They vary by size, by shape, by function. We have cells that are 3 or 4 feet long, the nerve cells that extend in the sciatic nerve from our hip down to our great toe is made up of single cells, each cell 3 or 4 feet long. Muscle cells are the size, in skeletal muscle, are the size of, or the length of the muscle. So, whether you're looking at your quads or your hamstrings or your calf muscle, your gastrocnemius, however large, whatever the length of that muscle is, its cells are that big. Other cells are small, they're all very tiny. Red cells and white cells, are both very, very small cells. They're sort of globular in shape as opposed to be long and stringy. So they have different functions, they have different shapes, they have different structures. Some cells don't have a nucleus. Red blood cells kick their nucleus out during development so we can have more room to carry oxygen. All the other cells in our bodies have a nucleus. Sperm cells have a nucleus and they have mitochondria and they have a flagella, and that's all they've got to kick everything else out, because sperm cells have one role, and that's to deliver the nucleus of the sperm to the female egg. That's all their role is. So they have cells are going to have different shapes and sizes and functions. Now, in a human cell, every human cell, regardless of its shape or size or function, is going to have the same three structures, except for red blood cells. They're all going to have a plasma membrane. That's the outer edge of the cell. So every cell is going to have a plasma membrane. The membrane is the barrier between the outside and the inside. It's like the skin of the cell. It restricts what can come in, it restricts what can go out. Every cell will have cytoplasm. That's the cell goop, or cell sap was written one time as describing it, like like a sticky, gelatinous, salty substance. And so that all the organelles are suspended in. And every cell is going to have a nucleus where our DNA is located. Now, red blood cells had a nucleus when they were developing, but right before they're ready to be released from our bone marrow, they get rid of their nucleus. So at one point, you can truthfully say every cell in our bodies had a nucleus. Red blood cells now don't have one. Well, you know, the red blood cells in our blood right now don't have a nucleus, at least they're not supposed to, because they kicked it out. So this is what a sort of a generalized cell looks like. We can see various structures on here. The largest structure, of course, is going to be the nucleus. The nucleus is this big purple thing right here. And it's surrounded by its own membrane called the nuclear envelope. That's the membrane of nucleus. The plasma membrane surrounds the cell all the way around. You've got various organelles inside the cell. You've got the mitochondria here, shaped like little sausage links. You've got structures, these green things are called Golgi apparatus. You can see other structures here called lysosomes. These are all the different organelles that all have specialized functions. And most cells in our bodies are going to have all of these organelles that do things for us. If nothing else, they're going to at least have a nucleus. Even a sperm cell has a big nucleus inside of it. Doesn't have a lot of other things here. It's not going to have to worry about making proteins or anything like that, but at least even a sperm cell will have a nucleus. Now, the cell membrane. Our cell membrane is a phospholipid bilayer. It's a phospholipid bilayer. It's made up of phospholipids. In other words, it is a fatty acid. It has two fatty acid chains attached to a glycerol, but instead of a third, it looks just like a triglyceride, except in place of the third fatty acid chain, it has a phosphate. And so the phospholipid looks something like this. You know, here is the structure of a phospholipid. This is the phosphate head. And here are the two fatty acid chains. So instead of looking like a typical triglyceride, which would have alcohol, you know, have the 3 carbons of glycerol and then have the fatty acid chains, there's your typical triglyceride. Here we kick off that third fatty acid chain and instead add phosphate to it, and we get something that looks like this. And this is fat soluble, but hydrophobic. These phospholipids hate water. And our phospholipid bilayer looks something like this, where we form an outer layer and an inner layer with fatty acid chains in the middle. And this is our lipid bilayer right here. This is the outside, and this is the inside. So our cell membrane is a double layer. We refer to it as the phospholipid bilayer because it's 2 layers. And all of our cells are going to have the cell membrane made of this structure. The nuclear membrane around the nucleus is made of this structure. In fact, the coating on all of the organelles within our cell itself is made up of these phospholipid bilayers. There are all sorts of things sticking up from the cell membrane. We have sugars, we have carbohydrates, we have lipids, we have, I guess a sugar and a carbohydrate is the same thing. We have lipids, we have proteins. We have proteins with lipids attached to them. We have carbohydrates with proteins attached to them. A lipid carbohydrate structure is very common on the surface of the cell membrane. And the cells themselves are going to be attached to each other. So the cells in our bodies aren't just randomly moving around. They're usually held together in some larger structure, and the cells have very specific junctions to hold them together. So they work as a large unit. Okay, here's what our, let's see how this is.
The plasma membrane is
There we go.
selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions, such as sodium ions and hydrogen ions, cannot pass through the membrane unaided.
Now, our phospholipid bilayer is very, very selective. We say it practices selective permeability. So certain molecules can come through and certain ones can't. As we saw here, oxygen and carbon dioxide have no trouble going across the membrane because they're very tiny molecules. Large molecules, glucose, for example, is too big. So glucose has to have help. Water can't get through the fat layer. So water has to have help. So the cell membranes of all of our cells can very easily restrict what can come in and what can come out. So there's your phospholipid, a little better drawing than mine, but it's essentially the same structure. The 3-carbon alcohol, you see the glycerol sort of tucked away there in our there's our glycerol sort of tucked away right there. Carbon, carbon, and carbon. There's 3-carbon alcohol. Here are 2 of the fatty acid chains here and here. And this is the phosphate attached on that 3rd carbon of glycerol. And so the phosphate head is what forms when you have the phosphate attached to the glycerol and you still have the fatty acid tail. So we have the head and the tail region, and the whole thing is hydrophobic. Water can't get in. So, and these fatty acids there's a double bond in here, right there. It has a kink, which is why this stays flexible and soft and squishy. It is not going to be a rigid surface. We're not going to have a hard, waxy, fat, solid fat surface here on the cell membrane. It's flexible. It's squishy because of the kink caused by the double bond in there. It's unsaturated fat. Get rid of the drawing. There we go. Now let's go on. And the layer in our membranes, the phospholipid bilayer, looks like this. A little better drawing than mine, but there's the phospholipid bilayer. We have the outside at the top and the inside at the bottom. And so what's outside has a hard time getting in through the barrier, and what's inside has a hard time getting out. Now, the things the cell wants, like oxygen, is going to go right through. The things the cell wants to get rid of, like carbon dioxide, are going to go right through the layer. It's very easy for the cell to do that. The things the cell needs, like water and glucose, the cell has a way to get those substances across the barrier. The same with ions like sodium and potassium. Things the cell needs to function. It will get them, even though the barrier would normally keep them out. It has special mechanisms that will allow certain ions to come through. It will allow water to come through. We have a way to get glucose into the cell, because those are all absolutely essential for the cell to survive. Now, this is an even more detailed drawing of the cell membrane. Here we see surface of a cell. It is, it covers the entire cell. Regardless of the shape of the cell, maybe it's one of those 4-foot long nerve cells running down your leg. Well, that nerve cell is going to have this same type of membrane all the way around it. Every cell in our bodies has this type of membrane. And it's in motion all the time. It's like a water balloon. Water balloon, you hold the balloon and you pick it up and it's moving around, and you lay it down and it's moving around. Imagine a water balloon that you could coat in oil and then roll in a bowl of Lucky Charms. And imagine the Lucky Charms then are floating on the oil and sliding around the surface. I picked Lucky Charms because it has all the different shapes there, because we see lots of different structures here in the membrane. Imagine this great big water balloon, and all the Lucky Charms, the cereal and the marshmallows are floating in the oil, sliding around constantly as the big water balloon moves. That's what's happening with the cell membrane. All these things on the surface of the cell are constantly moving up and down and sliding around in here. This is not a fixed, rigid, stationary structure. It is constantly in motion. So what do we see here? Well, of course, we see the phospholipid barrier here. We see the phosphate heads here in the outer surface and here in the inner surface. We see the fatty acid tails in between. We also see proteins that are embedded in the cell membrane. Here is a channel protein. Come on, there we go. Here's a channel protein right here. That's a channel protein. It's a simple passageway for water that comes through. It's a pipe. It's a tube into the inside of the cell or to the outside. Water can come through, or sodium can come through, or potassium can go out. Channel proteins are just essentially a tube in the membrane that substances can go through, just from one side of the membrane to the other. Over here, we have another what looks like a channel protein, but this may be what's known as a carrier protein. The carrier protein is a little more sophisticated. The carrier protein will limit what can go through. It may let sodium can go through. Sodium, yes. Potassium, no. Sodium can go out of the cell, but potassium can't. Or potassium can go into the cell, but sodium can't. The carrier proteins are a whole lot more specific. In fact, carrier proteins deliver glucose to our cells, sugar to our cells. Insulin that our pancreas makes when we eat something with carbohydrates in it and our blood sugar levels go up, we produce insulin. Insulin triggers the production of more carrier proteins in a cell membrane so that our cells can suck up more glucose, more sugar, and our blood sugar goes back to normal. I mean, we all know about insulin. Insulin's released from the pancreas. It's a classic negative feedback example. In negative feedback loop, we reverse the stimulus. Well, the stimulus is our blood sugar is up, so we release insulin and sugar goes into the cells, and our blood sugar level goes back to normal. we stop releasing insulin. Insulin works by by telling the cell to make more carrier proteins to let glucose come in, because glucose is a big molecule. It's too big to slide across the membrane. It's too big to fit through an ordinary tube. But a carrier protein, which is designed to carry glucose, can be built. And that's what happens. Insulin tells the cell to make carrier proteins. And we do that, and the cell gets all happy. It's getting its sugar like it wants. And then once we're done, then the insulin levels drop, the sugar levels go back to normal outside the cell. We stop releasing insulin. We stop making carrier proteins, and the carrier proteins that are there go away until we're ready the next time. And everybody's happy. So we have ways to get stuff into and out of the cell. These other things that are sticking up here, like these little green things, are called, this stuff here, this is called the glycocalyx. I'm sorry, no, this is called, this is a lipid sticking up here. This is called the glycocalyx. Glycocalyx. Now, what's a glycocalyx? I know you're just dying to hear. The glycocalyx is the identifier for us that tells the cell who we are. It tells the body who we are, so our immune system doesn't attack us, so that our immune system can recognize the cell as belonging to us, because every cell in our body will have glycocalyx structures announcing us as me. And so our immune system won't attack them. But if you have a transplant, if you have a foreign substance introduced into your body with foreign glycocalyx structures, then your immune system will attack it and try to destroy it. So it's very important to us that all of our cells carry these identifiers on here. And so while that simple little protein and carbohydrate linked together doesn't seem like much, it's very important to us. And this thing that says cholesterol right here, cholesterol is what we use inside our cells, among other things. But primarily we use cholesterol as reinforcing structures. It's like the rebar that goes into concrete. It makes it stronger. Cholesterol makes the cell membrane stronger. So another use for cholesterol. Yeah. So our barrier, our phospholipid bilayer, is 75% phospholipids, 5% glycolipids, fats and sugars, and 20% cholesterol. So the glycolipids, they're like a sugar-coated fat. Think of a funnel cake, if you will, because that's what, you know, when I think of a sugar-coated fat, that's what I'm thinking of. We put them, the glycolipids are on the outer surface of the membrane. We use them for signaling to other cells. We use them to attract other molecules to the cell. And then, of course, we have our glycoproteins, the glycocalyx structure, the protein and sugar. Okay. So I mentioned these proteins. The sugar-coated protein functioning as the glycocalyx, the carrier protein and the channel protein that allows substances to slide through the membrane. We have lots of proteins embedded in our membranes. Proteins allow the cell to communicate so the outside and the inside can remain in contact with each other, despite the fact that things won't go across the membrane easily. We have lots of membrane proteins. And the membrane proteins come in two types. They are what are known as an integral protein that extends all the way through the membrane, or a peripheral protein, which is usually found on one side of the membrane. And so some are free floaters, some are tied together, some go all the way through the membrane, and some just hang out on one side. And you can see how, and this is a really bad animation, but it's hard to get good animation for this type of thing. what you see here is the fact that the surface of a cell membrane is squishy, and it's moves up and down and side to side. The proteins are moving around inside the membrane too, whether it's an integral protein going all the way through or just a peripheral protein found on one side, like the underside of the membrane. Integral proteins go all the way through. They are what we call transmembrane proteins. They have, their job is to function as a carrier protein or a channel protein. I've given you examples of both of those. And they also function as a receptor site for other molecules to land on, or they function as an enzyme. We talked about enzymes the other day. So they may function as an enzyme for us to as a location for chemical reactions on the membrane of the cell to take place and do things for us. The peripheral proteins are generally found near the integral proteins. They also function as enzymes. They also can function as a what we call a motor protein. There are proteins that we use in our skeletal muscles to cause muscle contractions. And an example of a motor protein that we've seen would be the protein that causes cytokinesis to occur when a cell is ready. You know, it's ready to squeeze cytoplasm apart so it can finish the job of mitosis by having two daughter cells. Cytokinesis is squeezing of the cytoplasm, so we get these two daughter cells, and it's caused by a band of a motor protein that just squeezes and squeezes and squeezes. We call it a contractile protein. So enzymes, of course, for chemical reactions, motor proteins for muscle contractions and for cell division. And we also have specialized proteins for connecting between the cells, so the cells can communicate with each other. So these are all the peripheral proteins found usually on the underside surface, but not always, of the cell membrane. And this simple little slide here shows you all different types, all the different categories of the membrane proteins. I've already mentioned transport and enzymes and signaling and connections between other cell membranes and recognition and stabilizing the cell membrane. These are all different things that the cell does. We can see some of that here, for example.
Transport proteins carry molecules from place to place. The example shown here allows certain solute molecules to enter the cell. Hemoglobin is the transport protein that carries oxygen in the blood.
So we can have transport proteins in a couple different ways. One can be like a carrier protein or a channel protein that just opens up and lets a substance pass into or out of the cell. Or it can be a very large protein that floats in, that's found inside red blood cells that carries oxygen inside the cell. So we can see this working in two different ways, as a simple tube or as a very complex, large molecule that carries oxygen for us.
Receptor molecules bind to signal molecules and can then emit second messengers which trigger changes inside a cell. Receptors are thus important links in the system of communication among cells. Some signal molecules, such as hormones, are also proteins.
So what we have here, remember, the cell is very selective as to what comes into it and out of it. And some very large molecules are too big to even get through a carrier protein or a channel protein. They're just huge. And so they will land on a cell on what's called a receptor site for that chemical. That large molecule will land on a receptor site. It's too big to get in, but it will activate the production of a second protein to take a signal into the cell. We call it a second messenger system. So the large molecule can't get in, but it will land on a receptor and that will trigger a second molecule to move across the membrane smaller and tell the cell, hey, here's what you got to do. It works very nicely. Here we see structure.
Structural proteins have many functions. Like tent poles and ropes, they shape cells and anchor cell parts. They may serve as tracks along which cell parts can move. They bind cells together, making organized units such as muscles, ligaments, and the tendons that bind muscles to bones. The silk of spiders and the hair of mammals are also structural proteins.
So the structural proteins are essentially what help hold us together, to hold all of our cells together into an organized unit. And of course, this is enzymes.
An enzyme is a protein that changes the rate of a chemical reaction without itself being changed into a different molecule in the process. Enzymes promote and regulate virtually all chemical reactions in cells.
The advantage of an enzyme is that they are a biological catalyst. And since chemical reactions in our bodies are limited to functioning at body temperature with small concentrations and small particle sizes, the three conditions that we that control any normal reaction outside the body in a laboratory setting, we can't affect any of those three things. We're stuck at 98.6. We have small particles and small concentrations, but we can use enzymes to make those reactions occur rapidly. And the enzyme is a protein and it causes that reaction to occur, and it's not used up in the reaction itself and can go again and again and again to make a reaction occur. Now, this is the I want to take a few minutes and explain the glycocalyx here. I can't emphasize enough just how important the glycocalyx structures are to us. These are what we refer to as antigens. The glycocalyx structure are the antigens that identify us as self. The term antigen, A-N, self-identification. The sugar-coated protein, that sticks up on the surface of every cell, it is a glycoprotein. Every cell has a glycocalyx on it that is the identifier. It says, I'm me. The glycocalyx structure determines, tells us what blood type we are. All red cells have a glycocalyx structure telling us, excuse me, this happened earlier too. All of our cells in our bodies have a glycocalyx structure sticking up saying us, identifying us as us. Red cells have glycocalyx structures also telling us what blood type we are. All of our red cells have the same glycocalyx sticking up saying, hey, you're type A, or you're type B, or you're AB, or you're O. And that identifies us as our blood type. It identifies us as self. And so the glycocalyx structure is very, very important to us. It is our self-identifier. We call it the antigen. When we have an autoimmune problem, an autoimmune disorder, our immune system produces antibodies which will essentially attack those antigens. Now, normally antibodies are going to attack foreign antigens, but for some reason during an autoimmune response, we recognize the our own antigen, the own glycocalyx structure, as a foreign substance, and so we attack it, and that causes all sorts of complications for us. So we can just see how small the glycocalyx structures are. These microvilli are just folds in the cell membrane. And all they're designed to do, these are microscopic folds that are designed to increase the surface area of the cell. And the glycocalyx structures stick up on top of these little folds. So you can tell they are, you know, they are molecular, they are molecular in size because they're made up of molecules of proteins and sugars. Now, one of the problems we have with cancer cells is that while our body should be able to recognize the foreign antigen, the foreign glycocalyx on a cancer cell, when a cell becomes cancerous, it changes. And we should be able to recognize the cell as foreign because it has mutated itself. It has become something other than us. The problem is that our immune system takes several weeks to respond to a foreign antigen. It isn't going to be, for most things in our bodies, it isn't going to be an immediate response. Blood mismatching of blood is something different, but we'll talk about that in AP2. But for most cases, when we're exposed to a foreign antigen, we will produce a response within about 10 days, and we'll have lots and lots of antibodies formed to attack the foreign substance. And that's how we get rid of most infections. Bacterial and viral infections work, you know, that's how we get rid of them. If you get the flu, you're going to be sick for about 10 days, and then you're over it, because it takes that long for the antibodies to get produced to destroy the flu virus. If you were exposed to chickenpox, and some people still are, if you were exposed to chickenpox virus, it would take about 10 days before you start feeling better, because it takes that long to produce antibodies. Well, cancer cells don't play by the rules. Cancer cells keep changing, mutating themselves, so that they are always producing new glycocalyx structures before the immune system can recognize it as foreign. So if after 10 days or so, when you say, I've got it figured out, I can hear my antibodies for that particular antigen, well, the antigen's changed. It's totally new, and the immune system has to start all over. The immune system is always lagging behind here. Okay. Now cells, you know, as I said, you know, they're all, most cells are not free. Most cells are fastened together to each other to give us a whole body structure. You know, red cells float free, white cells float free. The 5 different types of white cells, the red cells, the platelets are all moving individually in our bodies. But most of our cells are attached to other cells, and they're bound together in 3 different ways. The cell junctions hold the individual cells together to form larger structures, to form tissues, and then tissues form organs, and organs form organ systems. And so the 3 ways that cells are held together are something called tight junctions, desmosomes, and gap junctions. A tight junction, we use the integral proteins, the one that span the membrane, to fuse together between 2 cell membranes. To fuse the 2 cell membranes that are side by side together, it's almost like they are stitched together on a sewing machine. You can see it almost looks like stitching down here. Where it's almost like it's sewn together, where the membrane, where the protein from one side of the membrane goes through the first cell membrane into the second cell membrane and is fastened off. So it goes back and forth like that. Tight junctions are found where we want to prevent things from getting into the cell or getting out of the cell. The cells that make up our wall of the stomach, the epithelial cells that make up our stomach wall, are held together with tight junctions so that stomach acids can't leak out into the abdominal cavity. So where we have tight junctions, we're going to have great strength, and we're going to have a practically impermeable surface. And so things can't get through here. Second type, desmosomes. Desmosomes go together like rivets. They look like a zipper, but it's like bolts shoved through the cell membrane from one side to the other and then anchored down. As you can see, here are the rivets. Here are the desmosome proteins going across. And over here we have keratin anchoring those rivets, those keratin rivets down. We use desmosomes where we want to have great strength, but we also want to allow for flexibility. So we have a little bit of give between the cells here. They're held together very tightly, but there is flexibility. They can move. And we see this in the heart, because the heart has to contract. The heart has to have powerful contractions every 0.7 of a second. And the cardiac muscle cells are held together with desmosomes. So they're not going to pull apart, but they're also going to have great pull against each other. Because a cardiac muscle cell, every muscle cell in the heart has to contract to its fullest extent every time. Whenever the heart contracts, every cell contracts. Every cell contracts to its fullest extent. The third category are the gap junctions. There are passageways that open up between the cell membranes. These are called the gap junctions. They're these proteins called connections form tunnels that go from membrane to membrane. And it's like a little tube. Now, the cells are usually held together with desmosomes, but they have these gap junctions in there. So you have instant communication from one cell to another, because normally when a cell communicates, it goes up to the surface of one cell and over to the surface of a next cell, then down into the cell. But here we're going straight through from side to side. It's a much faster way of communicating. When we want to send electrical signals from one cell to another, we use gap junctions. And our heart uses gap junctions to communicate from muscle cell to muscle cell. So within 0.7 of a second, both the upper two chambers of the heart and the lower two chambers of the heart have both contracted and relaxed based on the rapid spread of electrical signals from the upper chambers to the base of the lower chambers and back up the outside. Every 0.7 of a second for 90 years, if you will. So gap junctions, rapid communication between cells. Okay, let's see, a little bit here on passive transport. Passive transport across a membrane. When we look at cells, cells don't want to spend any energy. They're like all of us. We don't want to spend a lot of money to do things. We want to, be as economical as we can in getting things done. Well, the cell is the same way. Anything it can do that doesn't require a great deal of energy, it's going to be all about. We're going to be all in favor of that. And so the cells love passive transport, moving stuff across the membrane at no cost, not having to burn any glucose, not having to use any ATP. And the passive ways we do that are through diffusion, simple diffusion, facilitated diffusion or osmosis, or filtration, movement simply across the membrane. Now, simple diffusion across the membrane, simply moving a substance across the membrane from high concentration to low concentration. That's all it is. Oxygen moves into the cell through simple diffusion. There's always more oxygen outside the cell in our blood than there is inside the cell. So oxygen will always move across the cell membrane very easily. Carbon dioxide always leaves the cell through simple diffusion. There's always more carbon dioxide inside a cell than outside the cell in the plasma. Carbon dioxide leaves. There's no cost for this. It's a free ride. It's like going downhill, because simple diffusion always goes from the high concentration to its lowest concentration down the concentration gradient from high to low. So it's like going downhill, you know, or coasting downhill. You know, it's like the old roller coaster analogy, where you get to the top of the hill, and then the ride down to the bottom is a free ride because you've used all your energy getting to the top. So passive processes, cells love passive processes. So and this will be the last thing I'll show you. This is simple diffusion. We added dye to a solution, to water, and eventually we have a high concentration near the top where the dye went in, and eventually we reach equilibrium. The water molecule, the movement of the water molecules spreads the dye around until equilibrium is reached. The dye didn't have to use any energy. It's simply moving from high concentration to low concentration thanks to the movement of the water molecules. The same thing would work in air with odors. And so it's a free ride. And this is simply simple diffusion from the high concentration to the low concentration down the concentration gradient. Okay, this is a good place to stop. It's also 12:30, and I'm going to get us out of here. I have to go set up the lab test for my AP2 lab. Anybody have any questions on anything? Okay, then I will get you out of here, and I will see you all next week.